So yeah, I'm an undergraduate senior biology major at Bucknell, and my research focuses on the genetic diversity and connectivity of Chasmanthium latifolium in Pennsylvania and how that affects the conservation status. So this project is in collaboration with the between Bucknell and conservation practitioners with a common goal to conserve native plant species. So at Bucknell, we're able to provide genetic data and analyses, while the Pennsylvania Natural Heritage Program and the Western Pennsylvania Conservancy are providing much of the legwork for field sampling, as well as expertise dealing with the species outside of the lab. And through this collaboration, we hope to yield a scientifically informed conservation status assessment for Chasmanthium latifolium, as well as for the species that Angela and Cheyenne were talking about and will be talking about. To give a little bit of background on the species, Chasmanthium latifolium is in the Poaceae family, so it's part of the grasses, and it's native to riparian habitats. So that means it's along river and stream banks, making it fittingly referred to as river oats commonly. And it reproduces rhizomatously and through wind pollination, and it's a perennial species. Chasmanthium is characterized by its large, flat, and drooping spikelets, which are quite unique and can make it pretty easily identifiable, and is popular in cultivation, so you may recognize it. It's native to the southern Midwest and along the eastern half of the United States, and throughout this distribution, it's considered secure. However, Pennsylvania represents the northeastern edge of the geographic distribution, and within the state, we see two disjunct populations, one cluster of populations along the Allegheny River, and one cluster of populations along the Susquehanna River. The Pennsylvania Natural Heritage Program has assessed the species as critically imperiled, but the state still has a tentatively undetermined status. So the Pennsylvania Natural Heritage Program's recommendation as critically imperiled has taken into account a few factors. One significant consideration is the declining state populations, and this is partially due to recent growth in agriculture, industry, and housing, which has eliminated some of the natural habitats that Chasmanthium once inhabited. As well, we see an alteration in flood patterns through the damming of rivers, and being a riparian species, this can have a significant effect. We also see invasive species have contributed to the population decline. Another consideration is the disjunction between the eastern and western populations in the state. This can pose an issue for gene flow between populations given the significant length between them. And it's also an edge of range species. So I've mentioned that Chasmanthium is at the northeastern edge of the range, but why is that significant? Well, we often see that edge populations are highly adapted to complex and dynamic environments. And so if we see gene flow, it could have a couple different effects. If we have gene flow between edge and central populations, indicated by 1 on the map, this could increase genetic diversity, which is what we want to see, but it could swamp edge populations with traits that are less adapted to the harsh environments that they experience. Another possibility is gene flow between edge populations, indicated by the number 2 on the map, and this could increase genetic diversity while also maintaining those traits for the environment, which would be a good thing. You could also see that edge populations are isolated and there's no gene flow in or out of the population. And this could lead to inbreeding depression, which would not be good for the species. We've collected tissue samples at all 10 of the remaining populations in the state, and at each population, we collected samples from 7 to 16 individuals. These would be samples from a leaf or from the seed spikelets, and in total, we have 155 individuals that were sampled from. We've developed 3 different hypotheses for what we may see through the genetic analyses. First, we could see that there's one subpopulation within the state, where there's high gene flow between all localities collected from. We could also see, or alternatively see, that there's 2 subpopulations, one along the Allegheny River on the western half of the United States, and one along the eastern half of the states on the Susquehanna River. Within the eastern half and the western half, we could see gene flow along each waterway, but we would not see much gene flow between the east and western half. And then finally, we could see 10 different subpopulations, where each locality that we collected from is very isolated and does not have gene flow in or out between localities. And so we're using genetic sequencing to better understand what's going on in the population. This provides information on genetic diversity, gene flow between localities, and the levels of inbreeding, as well as the population structure for the species within Pennsylvania. And we hope to use the results generated from these analyses to better inform conservation status. Following the field collections, we extracted DNA from each sample of plant tissue collected, and this was done through grinding up the tissue and using a standardized protocol to isolate and purify the DNA we got out of each tissue. And after extractions, we quantified the DNA using a qubit. That's a device in the lab that is commonly used for this. And we also used gel electrophoresis to assess the quality of DNA. For sequencing, we really want long strands of DNA, and a gel can provide information on that. At the top of this image here, this indicates long strands of DNA, while further down are shorter strands. And we do have longer strands, as you can see, and that's what we want. It provides higher quality sequencing data. And sometimes we didn't reach the quantity or the quality that we wanted through an extraction, so we had to re-extract and concentrate those individuals to increase the yield. Following extractions, we sent DNA to the University of Wisconsin-Madison Biotechnology Center, and there they generated genetic sequencing data using a genotype by sequencing approach, similar to what Cheyenne talked about. This is a reduced representation genome sequencing, so enzymes are used to cut DNA to known gene regions, and we can compare them using SNPs. SNPs are single nucleotide polymorphisms, and this just means it's any variants that we see within the base pairing of the DNA. And this can provide information on diversity and a way to compare the genomes of each individual. After sequencing, the data generated from UWBC is run through a program called iPirad. The raw data that we get from sequencing isn't easily readable, and iPirad will sort, filter, and cluster the data to a usable format for analyses. After sequencing and assembly, we used RStudio for genomic analyses. R is a statistical program that is used by many biologists. It's an open source software and is capable of handling large data sets, so it's very useful for genetic work. within those many different packages and that we can use, and we used a couple to better understand the genetics of the species. We were able to generate some very preliminary data earlier this week, and this only looked at 8 of the 10 native populations within the state. And to give you an idea of the size of data we're working with, we have over 500,000 gene regions that we're looking at and over 1 million alleles that we're analyzing. First, we looked at heterozygosity, which is a measure of genetic diversity, and a value of 0 indicates very little genetic diversity, while a value of 1 indicates high genetic diversity. And our observed heterozygosity was lower than expected, so it indicates very little genetic diversity within all populations across the state. Next, FST was looked at, which Cheyenne talked about, is the coefficient of gene flow, and an FST of 1 indicates large genetic differentiation, while an FST of 0 indicates no genetic difference. And we had an FST of essentially 0, indicating that there's no genetic differentiation between populations within the state, and so there's very high gene flow between all of the populations. Then we looked at FIS, which is the inbreeding coefficient, and an FIS of 1 represents high inbreeding is occurring, while an FIS of -1 represents outbreeding from the population, and a value of 0.15, of greater than 0.15, signifies that inbreeding is occurring. Our FIS value was 0.56, so it does indicate that we have high levels of inbreeding within populations in the state. Next, a principal component analysis, or PCA, was conducted, and this clusters individuals based on genetic similarity. And like you saw in Cheyenne's presentation, if you have genetically distinct clusters, you could see them outside, separate from the rest, but within our Chasmanthium data set, we see that they're all kind of clustering together as one genetic unit. So what does all of this mean? To recap, FST was low overall, so that indicates that there's very little gene flow. FIS was high overall and indicates that inbreeding is prevalent. Heterozygosity was lower than expected, indicating that genetic diversity is low in populations across the state. And the PCA indicated that all populations cluster as one genetic unit. So given the preliminary results, we can conclude that Pennsylvania has one metapopulation with low genetic diversity and high inbreeding. However, with further analyses and much more thorough analyses, we could find something different is happening. This has some implications for conservation consideration. Although there is a limited and declining state distribution, and those populations appear to be healthy and quite large. Given the genetic data, we could see that these populations are actually quite threatened, and that would be important when considering how to conserve the species. And moving forward, I have a lot more analyses to look at, and once analyses are finalized, we'll be working with the Pennsylvania Natural Heritage Program to develop a more informed conservation assessment, and we hope to present this data at the Pennsylvania Rail Plant Forum eventually. And with that, I'd like to thank our funding sources, the Pennsylvania DCNR Wild Research and Conservation Program was the primary funding source for this project, as well as Cheyenne and Angela's research. And funding also came through the David Bopey Endowment and the Bucknell University Biology Department, as well as the Botanical Society of America. Rachel Goad with the Pennsylvania Natural Heritage Program, and Scott Schutte with the Western Pennsylvania Conservancy provided much of the field expertise and carried out much of the sample collections. So that was a huge help to this project. And I'd also like to thank the Martine Lab at Bucknell University, specifically Tanisha Williams, who has assisted greatly in genome assembly and analyses, as well as Dr. Chris Martine for his provided research support, and Angela McDonald, who provided much of the initial assistance for the project and developed the extraction protocols used, as well as the pipeline used for data analyses.